So I hope that you take advantage of that and I'm not wasting my money. Okay? What's that? Nothing? I guess it was another joke. Okay. All right, so I'm going to spend some time today talking about enzymes, and I'm going to cover a fair amount of ground. I'll probably finish with enzymes today. If I don't, I'll finish on Tuesday. The exam will cover through enzymes. Okay? So wherever I finish with enzymes, that's where the exam will cover through. That probably will be today. It might be a little bit into tomorrow. Okay? Well, last time I made the case for how enzymes work. And if you recall, that case was that enzymes work by, the magic that they do is by lowering the activation energy of reactions that they catalyze. That lowering of the energy is a transient thing. It doesn't change the starting reaction of the molecules or the ending reaction of the molecules. So that the overall change in energy that occurs in an enzyme catalyzed reaction is identical to that of a non-catalyzed reaction. Okay? Enzymes can't do the impossible. They can't change energies of reaction, but they can change energies of intermediates, and that's how they do it. Okay. Enzymes, like any other substances, are affected by temperature. There's a great story that we used to tell about the scientists doing a study on fruit flies. Maybe you've heard this story before about the fruit flies being an ideal gas. Have you heard the story? Okay. So the scientist has the idea that fruit flies behave like an ideal gas, because with an ideal gas we're thinking of random motion and we're thinking of relatively constant motion and so forth, and that if you heat them up, they in fact do speed up. And so the scientist sits down and notes that, you know, at a very low temperature like that of a refrigerator, the fruit flies are fairly, you know, lethargic. They don't fly very fast. They don't fly very much. They kind of hang around. As you start raising that temperature, they start becoming more active. And in fact, they do get a lot more active until you get them to a certain temperature, at which point they cease to be like an ideal gas. That's when they die. Okay? So that fruit flies do behave like an ideal gas to a certain point. Well, I make that analogy because enzymes are the same way. Okay? Enzymes, in general, will have an ideal range of temperature at which they work. Okay? If we go above that temperature, we may start denaturing the enzyme. So we're going to lose some effects. We'll also see as we talk about enzymes that there are pH effects. There are all kinds of different considerations. And enzymes have ranges of all of these in which they actually do function. Well, this particular table here has really little to do with the temperature, but it has a lot to do with activation energy. So we can actually compare the activation free energy, that is the energy it takes to get that reaction started, between, in this case, no catalyst, a platinum surface, and catalase. Catalase catalyzes the breakdown of hydrogen peroxide. And we can see that, of course, in each case that the enzyme is lower. And that lowering of the activation energy has a dramatic effect. If you look at what catalase is doing, all right, catalase is increasing, okay, the reaction rate, the relative rate of this reaction by 650 million fold. All right? That reduction in energy really makes a heck of a difference. All right? It's like saying that you're going to climb Bald Hill versus Mary's Peak. Okay? Climbing Bald Hill is a relatively easy thing to do. Mary's Peak is another matter. Maybe if we did Bald Hill compared to Mount Rainier or Mount Everest. Okay? So there's some really big differences that happen. Even if as we lower that activation energy by a relatively slight amount, we see some tremendous increases in the relative rates of reaction. Okay. Here's the temperature effects I was thinking about earlier. We see that enzymes, not surprisingly, do in fact have a range of temperatures over which they work optimally. This is the percent maximum activity here on the y-axis against the temperature on the x-axis. And we can see that for this particular reaction, the ideal temperature is around 40 or so degrees. And not surprisingly, 40 degrees centigrade is around our body temperature. So most of our enzymes have an ideal reaction velocity around the temperature of our bodies, but not all. And we'll also see that some actually have some interesting variations as pH changes, and they're not always at what we would think of as physiological pH. All right. Well, as we think about enzymes and what they do, it's important for us to think about how they do it. All right. So how enzymes work. I've talked about how they lower an activation energy. I want to talk about another consideration in how enzymes work. And it's this consideration that I think and I hope will convince you that enzymes are very different from a chemical catalyst. Okay? All right. Well, what you see on the screen are two different models for explaining how an enzyme works. Okay? When we talk about a model, all a model does is a model explains a certain phenomenon. All right? It doesn't have to mean it's right. It's just that this is the way somebody has described it. This is the way that it works. That's what a model is. Okay? And so models provide a means of testing to see how they actually do work. And what we find in testing is that the models really tell us some interesting things. So the old model of how enzymes work is called the lock and key model. Okay? It's called the Fisher lock and key model. And that lock and key model is typically what's taught to students in basic biology, even in basic chemistry. I'm sure you've heard of the lock and key model. Everybody heard of the lock and key model? Yeah. The lock and key model says that the substrate is the molecule that the enzyme acts on. The substrate fits into an enzyme like a key fits into a lock. That there's a nice sort of handshaking thing that's there. They're perfectly fitted for each other, and that's how the enzyme does what it does. Okay? Well, that model turns out to be very good at explaining how a molecule is adapted to by an enzyme. That is, how it fits in there. But it's not at all good in telling us how an enzyme does catalysis. Okay? What this model says, what the Fisher lock and key model says, is that the substrate fits into the enzyme's active site. And by the way, the active site is the place in the enzyme where the reaction is catalyzed. It says that the substrate fits into the active site, and then magic happens and the reaction occurs. I'm exaggerating, of course, but it doesn't explain the reaction to us. Okay? Now, there's another model that's called the Koshland, K-O-S-H-L-A-N-D, the Koshland induced fit model, that really tells us a lot more about how enzymes work. All right? Now, it's a little hard to tell on the screen. The figures that people draw for these really aren't the best, but let me try to explain the differences of these models to you. The model on the left is drawn so that the enzyme has the exact shape of the substrate. You see the substrate has sharp edges. Those sharp edges fit into there, and everybody's fine and dandy. The model on the right is drawn to show you that the enzyme has something sort of like the shape of the substrate, but it doesn't have sharp edges. It doesn't fit exactly. And that's a very critical component of the induced fit model. I'm going to give you a sentence that tells you what the induced fit model does, and then I'm going to explain it to you. Okay? The sentence says that not only does an enzyme change the substrate. Okay, the Fisher model also says the enzyme changes the substrate because it's catalyzing a reaction, right? Not only does the enzyme change the substrate in the induced fit model, but the substrate transiently changes the enzyme. That's something that's not in the Fisher model. And that's a very, very important difference. Now, I'll tell you why that's important in a second, but I want you to understand what I just told you. Not only does the enzyme change the substrate, but the substrate transiently changes the enzyme. Okay? That contrasts with the Fisher model, the lock and key model that says, well, this guy fits in here and then everything happens and then the reaction occurs. It doesn't tell us how the reaction occurs or anything. So how does the induced fit model explain how an enzyme does what it does? Well, if the enzyme doesn't exactly fit the substrate to start, we can imagine that here's this enzyme, and it's got sort of a general shape of the substrate. So the substrate can sort of, it's kind of like a key you can pick a lock with, right? You can sort of get that key into the lock, and then you try to get the unlocking to occur. As that substrate comes in, the substrate has hydrogen bonds it makes with the enzyme. It might have ionic bonds it makes with the enzyme. It might have any kind of interactions with that enzyme. And just like we saw, hemoglobin had its structure slightly change when oxygen bound to it, so too does the enzyme have its structure slightly changed when the substrate binds to it. Okay? So that binding of the substrate is slightly changed in the enzyme. What does that do? Let's imagine here's my substrate and here's my enzyme. Okay? This guy binds, and now the enzyme is slightly changed in its configuration. That slight change in configuration may lead to strain. Okay? It may lead to strain. So for example, if this enzyme normally splits this molecule in half, this strain might cause a split in the substrate that's being bound. When the reaction is done, the substrate leaves and the enzyme goes back to the way that it was. That's what a catalyst does. It goes back to the original state. Now that's a little hard to understand example. I'm going to give you a little better example. There's an enzyme we'll talk about later called hexokinase. You don't need to know the name of that at this time. But I use this example to show you what it does, and I think this illustrates it really well. Hexokinase is an enzyme that catalyzes the first reaction in the breakdown of glucose. Okay? In the reaction that hexokinase catalyzes, it transfers the move of a phosphate from ATP onto glucose. So phosphate's moving from ATP onto glucose, and hexokinase catalyzes this. Now, what I'm going to show you, I hope, will illustrate that. I'm going to stand in front of the screen so that people out in TV land can see it. Okay? So here is the hexokinase enzyme. It's got two different places to bind things. One place up here binds ATP, and one place down here binds glucose. Okay? Binding of both of these, let's say glucose binds, and then hexokinase then ATP binds. The binding of the second one causes a conformational change in the enzyme. And here's what it does. The jaws close. Okay? Now that movement that I just showed you right here can happen inside of an enzyme because an enzyme is a protein, and a protein is flexible. A chemical catalyst is not flexible. It's called an induced fit because the shape change is induced by the binding of the substrate. Well, now you can see I've just closed the jaws on this. Now the ATP that was on the top is brought into very close proximity with the glucose that's on the bottom. It becomes very trivial for the phosphate to move from the ATP up here to the glucose down here. When that happens, there's a conformational change again. The jaws open. And now ADP and the glucose with the phosphate go flying away. Now that induced fit tells us how that enzyme catalyzed that reaction. That's one example. There are thousands. Okay? The Fisher lock and key didn't tell us anything. It just said it goes in there and it comes out and it's different. The induced fit model told us how that change occurred. Okay, questions about that before I go on? Yeah?
Would that be cooperativity like hemoglobin?
Would that be cooperativity like hemoglobin? In fact, it would not be cooperativity. In fact, in enzymes, we'll see a phenomenon called allosterism that affects enzymes kind of like cooperativity affects hemoglobin, but that's not an example of it. That actually is an induced fit change. It's actually a structural change, a transient structural change that's happening. Okay? Other question? Yeah?
That's called hexokinase.
The enzyme is hexokinase, yeah. And there's other examples of that as well. You don't need to know the enzyme name. Okay. So, blah. Okay. Now, let's think about other things about what enzymes do and how enzymes work. Okay? Chymotrypsin is an enzyme that catalyzes the breakdown of peptide bonds. I showed you trypsin. I told you trypsin cut next to lysines and arginines. We talked about that last time. All right. Chymotrypsin is an enzyme that also breaks peptide bonds. It's the amino acids it cuts next to are a little bit more varied, so we don't have to worry about that. But what chymotrypsin does is it breaks a peptide into, it breaks a bond right there. That is wrong. That should be a nitrogen right there. That should be a peptide bond right there, and it should be making an amine and an acid. Okay? Well, hexokinase turns out to like a lot of general structures. We don't see all enzymes only working on one thing. And hexokinase is kind of flexible. And we can imagine the more flexible it is, the more substrates it can accommodate. And that's what chymotrypsin, I keep saying hexokinase, that's what chymotrypsin does. Okay? Well, that turns out to be really useful because, as I said last time, biochemists are lazy people, and biochemists like to study things as simply as they can. It turns out that chymotrypsin will work on this guy right here, that even though it's not a peptide bond, it looks enough like it to chymotrypsin. The chymotrypsin will actually catalyze a reaction on it, and that turns out to be really useful for studying it. Why? Because when it catalyzes this reaction, it creates a product that is yellow. The starting material is colorless. So if I want to study chymotrypsin's reaction, all I have to do is watch how fast the yellow appears. Okay? Everybody with me? I simply have to watch how fast the yellow appears. A machine can tell me that very readily, and the faster the yellow appears, the faster chymotrypsin is working. Very simple. Okay. Now, this is another graph. I need to tell you how we do this graph and what we're moving in the direction of. We're very interested in studying enzymes to understand how fast enzymes work. I hope I've convinced you that the speed with which these enzymes work is very interesting. And it turns out that the speed with which they work is important to understand because cells have to control that speed. Okay? I'd like to give the example that if you want to go to Fred Meyer, you can go there in a Maserati, but somebody's going to have a speed limit because if you go there at 200 miles per hour, there's probably going to be some problems, right? Well, if we have an enzyme that is working uncontrollably and it's working at the equivalent of 200 miles per hour, it may be breaking down things faster than the cell can use them. It might be causing some problems. So cells really need to be controlling enzyme rates. It's for that reason that we study the enzyme rates in the laboratory a lot. All right. Well, what are we looking at here? Let's say I've got this enzyme. I've got chymotrypsin. And let's say I've got one chymotrypsin molecule or one chymotrypsin enzyme in a test tube. And let's say I've got 20 substrates. In this case, this big name molecule. We won't even worry about the name of it. Okay? Got 20 substrates. I got one enzyme. 20 in a tube is not very many, even if we have a very small volume. Okay? It's going to be a fair amount of time that the enzyme is going to bounce around before it finally bumps into its substrate. Okay? Because all of these are happening by Brownian motion, random movements, right? It's all that's involved in getting together an enzyme with a substrate is random movement. So the lower the concentration of the substrate, the less likely it's going to bump into the enzyme. Now let's contrast that with a very high concentration of substrate. Let's say I've got 20 billion of those substrate molecules and I've got one enzyme. We would imagine that it's going to be much more likely that one of them is going to bump into the enzyme, right? If it's a random process and I've got that many more, it's going to be much more likely that one of them is going to bump into the enzyme. All right. Well, that's what this experiment is showing you. So it's showing me the following. I'm interested in studying how this enzyme is affected by the concentration of the substrate. So what do? I take, in this case, they show about 6 different points. Typically, I would do about 20. I would take 20 different tubes, and into each tube, I would do the following. I would put some buffer. Okay? I would put, in tube number 1, I would put in a certain concentration of substrate. In tube number 2, I would double it, or something like that. Tube number 3, I might make 5 times as much. Tube number 4, I'd make 10 times as much. What you see I'm doing is I'm varying the substrate concentration in each tube. Increasing amounts of substrate as I go along. Then, I take and I put a fixed amount of enzyme into each tube. A fixed amount. All right? Why? Because I only want to have one variable in an experiment. If I'm varying the substrate concentration, I don't want to be varying the enzyme concentration as well, because I can't make sense of that. So now I've got 20 tubes. Each has the same amount of enzyme. They each have the same buffer, same pH, everything else. And I let each one react for exactly the same amount of time. And at the end of the reaction, in this case, I measure the amount of yellow. Yellow is the reaction velocity. The more yellow I have, the faster the reaction is going. Okay? Well, when I plot that, I say, okay, tube number 1, here was the concentration of my substrate. Here was the amount of yellow I got. Tube number 2, here was the concentration of my substrate. Here was the yellow I got. Tube number 3, etc. Okay? What I generate is what we describe as a hyperbolic curve. Now you'll notice it's a curve. It's not a straight line. There's a very good reason why it's not a straight line. I'll explain that to you in a second. But you notice that it's a curve, and it's called a hyperbolic curve. It flattens off. It flattens out. It will eventually reach a point that we'll call maximum velocity. Maximum velocity. Now, maximum velocity is an important parameter in studying an enzyme. That's the place where that curve levels off. It's going to level off at maximum velocity. Well, why is there a maximum velocity? Let's imagine that I'm making automobiles for General Motors. Okay? And I get all the parts together, and I've got one factory that does all the assembly. If I start making cars and I have limiting numbers of mufflers, I can only put out a complete car as fast as I can supply it with mufflers. Right? Well, then I'm the boss and I say, we've got to increase production, so get me some more mufflers. Everything else is abundant. Give me some more mufflers. I start increasing numbers of mufflers. What's going to happen? Well, that factory is going to start putting out more and more cars, but eventually the factory is going to produce a maximum number of cars. Why? Well, there's only a certain number of workers there. There's only a certain amount of machinery that's there. A factory has a maximum capacity, right? An enzyme has a maximum capacity, and we call it maximum velocity. Okay? Makes sense? Yes?
Over the course of one, because these are averages of different concentrations.
Yep.
Over the course of one concentration, wouldn't it be more like a parabola or something?
Well, I'm not sure I understand your question. Over one concentration, meaning what?
So if you take...
So this would be the average of 2 or 3 runs of this concentration. So I might see values ranging across here.
Right. I'm saying over the course of the enzyme acting upon the substrate,
Uh-huh.
and it creates more of the products.
Yep.
So that would change the concentration of the substrate. So we can...
Oh, you're asking a very good question. So let me come back to that, okay? Because what she's saying is, as I start using the substrate, I'm changing the substrate concentration. And she's right. Okay? So let me briefly say this. What we do when we measure these is we measure what's called initial velocities, so that we don't get a chance to change the concentration very much. But it's a very, very good point that you're making. Okay? All right. So maximum velocity is like the factory's maximum capacity. Now, there's a very important point, and if you guys understand this, you will understand something better than a lot of biochemistry majors understand. Okay? Maximum velocity is not a constant for an enzyme. I know professors who think it's a constant for an enzyme, and it absolutely is not. Let's think back to General Motors. The boss says, I got you all those mufflers. You went from making four cars a day to making 500 cars a day. But now I got plenty of mufflers, and no matter how many mufflers I give you, you still make 500 cars a day. If the boss is smart, he's going to open another factory, right? If I want to make maximum velocity higher, I simply have to add more enzyme. Maximum velocity depends on the amount of enzyme. It's not a constant. It depends on how much enzyme I use. Just like factory capacity depends on how many factories I have. Okay? Yeah. Temperature and pH can affect things, yes, but we're doing all these at the same temperature and pH because we have a fixed buffer, fixed concentrations. Yep. Okay. Now, so maximum velocity depends on how much enzyme I use. It's not a constant for an enzyme. I can't say the maximum velocity for hexokinase is blah, 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 because the first question I have to answer is, well, how much enzyme did you use? We want to have a parameter that is independent of the amount of enzyme that we use. And it turns out it's very simple to do. We take the maximum velocity and we divide it by the concentration of enzyme that we used. And now we've taken the amount of enzyme out of the equation because it's canceled out with the concentration of the substrate. The two concentrations actually cancel. So if I take the maximum velocity and divide it by the concentration of enzyme that was used in that experiment, I get something called kcat. K with a subscript CAT. That stands for catalysis. More commonly, we call that number the turnover number. And you'll see why in a second. The turnover number. The turnover number, first of all, is a constant for a given enzyme as long as we have a given set of pH and so forth, temperature and so forth. It's a constant for a given enzyme. It tells us something really, really simple. Okay? You've actually already seen it once so far this term. Okay? I think that maybe I'm confusing. I always forget which term I'm in. So I'm an absent-minded professor, right? All right. The turnover number is a number. If I have something that has a turnover number of 1,000 per second, that might be what a turnover number might be. What it means is that every molecule of enzyme is making 1,000 molecules of product per second. 1,000 per second means that every molecule of enzyme is making 1,000 molecules of product per second. There are some enzymes that have turnover numbers of 1,000,000 per second. That's a phenomenal number. How can 1,000,000 things happen per second? But they do with some enzymes. Okay? The turnover number is a constant for a given enzyme. Vmax, I should say maximum velocity is also called Vmax. I didn't tell you that. Maximum velocity is called V with a lowercase max. Vmax is not a constant for an enzyme because it depends on the amount of enzyme that we use. Okay, questions? Yes, sir.
So is that like a ratio of how much substrate to enzyme it has?
So what the turnover number is in fact a ratio of substrate to enzyme. That's correct. So that's why when the numbers say 1,000 per second, that is the ratio of 1,000 molecules of product per molecule of enzyme per second. And the turnover number tells us that number. Okay. Good, good. Very pleased that you guys get that. I'm going to come back and talk about ATCAs in a second. All right? We don't need to worry about first-order kinetics and second-order kinetics. That's not something that's really a part of our course. But this does show us, okay, something that looked very much like what the last figure. In fact, it's identical to the last figure. But now we see a more explicit labeling. Velocity on the y-axis versus substrate concentration on the x-axis. We call this a V versus S plot. V versus S. V standing for velocity, S standing for substrate concentration. Okay. Velocity versus substrate concentration. V versus S plots for many enzymes will give us a hyperbola just like this. Not all enzymes, though, will do that. That's what ATCAs does. If I look at ATCAs, it's another enzyme that catalyzes another reaction. Here's what it looks like. It doesn't look like a hyperbola. In fact, it looks like something that you've already seen. What does it look like? It looks kind of like, well, it's not quite a titration. No, there's something else you've seen that looks like this. Hemoglobin with oxygen, right? We saw hemoglobin. Remember that we had myoglobin that looked hyperbolic, and hemoglobin had that S shape to it? Okay? This S shape tells us that this enzyme has other considerations. What caused that S shape in hemoglobin? What was it that was causing hemoglobin to change? Oxygen was changing it, right? So you asked about cooperativity earlier, all right? That cooperativity was seen when the first oxygen bound, it changed the enzyme, and the enzyme started binding oxygen more readily. That's why hemoglobin had a sigmoidal shape. This enzyme has a sigmoidal shape, and you might think, well, does the binding of one substrate affect the binding of additional substrates? And it does. So just like hemoglobin had a cooperative effect, this enzyme has what we call an allosteric effect. A-L-L-O-S-T-E-R-I-C. I'm going to define that for you in a second. Okay? Allosteric effect. Allosteric effects are what we talk about with respect to reactions being catalyzed. Cooperativity is what we call essentially the same phenomenon, but there's no reaction being catalyzed. Okay? What does allosteric mean? Okay? We refer to the phenomenon as allosterism, right? Allosterism, allosteric effects, same thing. Okay? Allosteric effects occur when a small molecule binds to an enzyme and affects its activity. A small molecule binds to an enzyme and affects its activity. This turns out to be really important because it's through this mechanism that's one of the ways in which cells can control an enzyme. There are other ways of controlling enzymes. This is one of them. Allosteric effects, or allosterism, same thing. Now, there's a lot of stuff we're going through here, right? Let's ponder that for a second. Questions? Maybe everybody stand up. I think you guys need a break. Stand up. Stand up. I want everybody to take a deep breath. Okay? And we're going to go jog that half marathon. No? Jumping jacks? Ready? No? Somebody say something non-biochemical. What? I didn't hear what you said. Booger, okay, well, all right. I think that is biochemical, but uranium's not. Okay, I have a stupid joke for you. How's that? Have a seat. That was kind of abrupt, wasn't it? Everybody knows, of course, that fishies like to swim in schools. Little fishies like to swim in schools, right? What's their favorite subject? Nobody knows this. What's their favorite subject in these schools? Debate. Debate. Okay. All right. That's said. All right. I didn't feel like you had enough concepts, and now you need some more. This figure doesn't do a very good job of showing us Vmax because it looks like it's a long ways above there, but they're assuming this is just going to eventually get up and interact with that. Vmax actually, as I said, it's important. It varies depending on the concentration of enzyme we use, but for a given enzyme, it will be a specific Vmax. And the reason that we have a Vmax is because the enzyme has become saturated with substrate. Saturated. That's what happened with the factories. You got more mufflers than you can handle. There's still a limit in how fast you can put things through, right? The enzyme is saturated with substrate. It doesn't have to wait for substrate to get in there. There's always another one waiting. It's always ready to go. Okay? So Vmax, and more importantly, kcat, tell us a lot about how fast reactions go. But there's another parameter that we think about with enzymes that tells us something about how much the enzyme likes its substrate. How much does it want to bind to it? What's its affinity for its substrate? Km is more like the things you think about with your significant other. Right? If you have great affinity for your significant other, I'm sure you've seen many public displays of affection that are really disgusting. But people have different affinities for their significant other, right? Enzymes have different affinities for their substrate. And we measure that with a parameter called Km. Km. That's the thing down here. I'm going to explain it to you in a second. Km is a measure of the enzyme's affinity for its substrate. An enzyme can have a high affinity for its substrate. It can have a low affinity for its substrate. How do we measure it? Well, you can see on the thing on the screen that Km is a substrate concentration. But you'll note that it's not the substrate concentration at Vmax. Why don't we use Vmax? Vmax is saturated. So once we get saturated, we could have an infinite number of those, right? It doesn't tell us anything about the enzyme's affinity. It's kind of like having, I don't know, I hate it when I don't think of an analogy before I start opening my mouth about it, but here I am doing it. It's kind of like having 50 students in a class, which is about what you can handle reasonably, versus 500 students in a class. When people come to see you at office hours, right, you're going to have just about as much likelihood of catching the professor with 50 as you are with 500, which is going to be low, right? You're going to be waiting in line. They're both going to be very difficult, very limiting for you to get, all right? So we don't measure Km at Vmax because there would be all kinds of possible things out here. Instead, we measure it at Vmax over 2. Vmax over 2. Yes?
How do you apply Vmax to a sigmoidal curve?
How do I apply it to a sigmoidal curve?
Yeah.
I mean, how do I decide what this line is?
Yeah, I mean,
Yeah.
What do you
Oh, to a sigmoidal. A sigmoidal will also flatten off. I'm sorry, I wasn't thinking what you were asking me. It will flatten off also, but for our purposes, we won't need to worry about that. But it'll flatten off very similarly. It just takes a little while getting there. Okay? Okay. Well, once I've determined Vmax over 2 becomes pretty trivial to determine. And this turns out to be really useful for us. If I measure the substrate concentration that it takes to get to Vmax over 2, I have just determined what Km is. Km is a substrate concentration. It is that substrate concentration that causes the enzyme to get to Vmax over 2. I'll repeat that. Km is the substrate concentration it takes to get the enzyme to Vmax over 2. Why does that tell us anything? Well, let's imagine I've got 2 enzymes. They both work on the same substrate. One really has a great affinity for its substrate. Wherever it sees it, it grabs it and gets a reaction. This guy over here is like the absent-minded professor, kind of like me. Okay? It takes a fair amount of substrate pounding it in the head before it finally wakes up and goes, oh, you want to react. High affinity, low affinity. Okay? Which one's going to take less substrate to get to Vmax over 2? High affinity. It's going to be grabbing it, right? It's wide awake. It's alert. It's doing its thing. Okay? High affinity means low Km. And conversely, low affinity means high Km. It takes a bunch of substrate pounding it in the head to make it wake up and start reacting. There's an inverse relationship then between affinity and Km. High affinity, low Km, low affinity, high Km. Okay. Yes, question.
You said Vmax is a constant, right?
Vmax is not a constant, no. It's a constant at a given concentration of an enzyme.
This one is Km constant?
Km, that's a good question. Km is a constant for an enzyme under a given set of conditions, yes. Okay? It's a parameter.
Given set of conditions, you have a set Vmax and a set Km. Change the conditions, you get 2 different.
Okay, so her question is, if I have a given set of conditions, I'll have a given Vmax and a given Km. Well, not quite. If I change the concentration of enzyme, I will change the Vmax, but I won't change its affinity for its substrate. So the Km will stay constant no matter how much enzyme I use. It's a very good question. Thank you for asking. Okay, Zunay. Yeah. So Km is a constant for an enzyme. Okay, given a set of conditions, right? And that doesn't matter how much enzyme I use. The affinity, if you think about it, that affinity doesn't really matter, right? I wouldn't think affinity would change if I changed the amount of enzyme, because the affinity is an interaction between the enzyme and the substrate. So I would expect that when I measure affinity, I should get a constant value, because that's going to be true no matter how much enzyme I've got. Whereas velocity, which is a function of how many enzymes I have, is going to vary with that. Does that make sense?
Yeah.
Good, good. Other questions? Yes?
Are we going to learn what determines affinity?
Are we going to learn what affinity is? Is that what your question?
What determines affinity?
Oh, okay. What determines affinity? We're actually going to see some things that will affect affinity. So that's actually a little further along. There are things that will affect affinity. Okay? One of them is molecules that interact with the enzyme. Enzymes are affected by things that interact with them. Enzymes are also affected, and you don't need to know this, I'm just telling you because she's asked, okay? They're also affected by chemical alteration. We see enzymes get chemically altered by cells. That's another way to control them. That will also affect their ability to, or their affinity for their substrate. Yes, ma'am?
Experimentally, how do you find your Vmax over 2? If that's your way of measuring the affinity,
How do you determine your Vmax over 2? Very good question, okay? So I know how to do a V versus S, right? I use 20 different tubes. I've got my varying substrate concentration, constant amount of enzyme, right? That's going to give me Vmax. So now I know Vmax. I just divide it in half, and I've got Vmax over 2. Right? Good question. Yeah. You want one more thing, or you want to save it for tomorrow? Yeah.
When did you say your TA's out for?
I didn't say. I did email them out, and I'm going to put a link on the class webpage where you can see those, and you can come and talk to them as well. Okay. So what? I'm sorry. Question? Oh, okay. What I was going to say is I do want to finish enzymes for the exam, so I can either get pretty much through that today, or we can tackle it tomorrow. Whatever. How many want today? We're not going to finish it today, no matter what we do. Okay. The question is, do you want one more topic, or do you want to have a topic tomorrow? All right. You guys look tired. We'll have it tomorrow. That means we'll have the song tomorrow too. Okay. Yes, sir.
This is probably a dumb question, but if you've got a